What's up, everybody? Today we're going to handle aerobic respiration. Aerobic respiration is going to be what we utilize in our cells to completely break down sugars. We already talked about glycolysis, which is an incomplete breakdown of sugars. This process is going to be used in both prokaryotes and eukaryotes, and it requires oxygen. Okay, so that's what this word means, aerobic. It's in referring to the use of oxygen. Anaerobic means without oxygen. So if we put an AN in front of it, just in case we see that later on. And of course, fermentation would be an anaerobic process. All right, so let's get into it. So as a result of glycolysis in the cytosol, we have an accumulation of pyruvates. So the first thing we're going to look at is in prokaryotes, what's going to happen with these pyruvates, okay? And we already talked about this pathway here. If there's no oxygen present, we know that a prokaryote is going to make alcohol in a process called fermentation, and as a byproduct of that, it's going to be releasing carbon dioxide, okay? But what we want to focus on here today is oxygen. So if we have oxygen around, what's going to happen is that pyruvate is going to be able to be metabolized, broke down completely, okay? So first thing that's going to happen to it is the transition reaction, and then that's going to be followed up by the citric acid cycle. All of these processes, the formation of pyruvate through glycolysis, the transition reaction, and the citric acid cycle, they're all taking place in the cytosol or the cytoplasm. So I tried to depict here like a bacillus, a rod-style bacteria, and in the cytosol would be where all of these initial reactions take place. Electron transport chain is going to utilize several proteins that are embedded in a membrane, and that's really where the vast majority of ATPs are going to be produced. We're going to look at this in more detail later. It's an identical process, really, all of these between the two prokaryotes and eukaryotes, except for where they're occurring. But electron transport chain occurs in the cell membrane, so all those protein complexes are embedded in the actual cell membrane of a prokaryote. Okay, so in terms of a eukaryotic cell now, the pyruvates that accumulate in the cytosol, if there's no oxygen present, again, we're going to go through fermentation pathways. And we know that this can vary depending on the cell. So if we were looking at like a fungal cell, like a yeast, right? Or if we were looking at a plant cell, like barley or grapes or anything like that, we're going to go through alcohol fermentation, okay? If we were looking at an animal cell, what would happen is the cell would go through lactic acid fermentation, which we've already discussed, okay? So again, we're going to focus here on what would happen if oxygen is available. And for right now, what I want you to understand is that if oxygen is available, these pyruvates are going to actually leave the cytosol and go into this organelle, which is called a mitochondria. All right, so we're going to get into some more detail about this organelle. It's a very important organelle for life. So mitochondria came to exist in eukaryotic cells about 1.5 billion years ago. Right along line with our first eukaryotic cells is what most of the modern day theory is surrounding this. And what happened in the theory of endosymbiosis is that prokaryotic cells were being engulfed, consumed, if you will, by other cells. And these cells in particular, or this organelle, came from an ancestral cell that instead of being digested was able to exist within the host cell, if that makes any sense. And it really has tremendous functionality. It's able to break down sugar, really the products of glycolysis. It can break them down in a more complete manner. Okay, so this organelle has all of the machinery that an ancient prokaryote would have. It actually has its own DNA, its own genetics. And that DNA codes for protein complexes, just like I tried to show you earlier in the prokaryote, that are actually embedded in the cell membrane here, okay? So in order to understand how this thing works completely here, we need to make sure we understand some anatomical structures found within the mitochondria. So this first membrane is called the inner membrane. And of course, the mitochondria, just like many organelles, have a double membrane, similar to a chloroplast. So this outer membrane is out here, surrounds the organelle, and there's a space in here, a gap between the inner and outer membrane. We call this the inner membrane space. And then inside of that, this is called the mitochondrial matrix, which I depicted here in yellow. Okay, so the inner membrane space purple, matrix yellow. And I mentioned it briefly, but I just want to go back and revisit the fact that mitochondria have their own unique DNA signature. This is inherited in the maternal line in human beings. So you have your mitochondrial DNA came directly from your mother, okay? And hopefully everybody's with me there. All right. So what's happening is the pyruvate is entering into the mitochondria. It's going into the matrix. Once inside the matrix, the pyruvates, the 3-carbon pyruvates, are going to get converted into 2-carbon acetyl-CoAs. So I hope that it's clear that we're losing a carbon here in this process. It's called the transition reaction, and we're ending up with 2 acetyl-CoA molecules as the product. Okay. So as a byproduct of this exchange or this transformation, we're going to lose carbons to the environment, and those CO2s get put into our blood. That's the waste product of this transition reaction. It's the first time that carbon dioxides are given off in any process involving cellular respiration, so it's worth note. But this carbon dioxide enters the bloodstream and is converted into carbonic acid, and then later it's going to be expelled when we respire, turned back into a gas in the lungs. Okay. So this is the transition reaction again in the mitochondrial matrix. The next step of the process is called the citric acid cycle. In some textbooks, they're going to refer to it as the Krebs cycle. And these two terms are synonymous, mean the same thing. But what's going to happen is those acetyl-CoAs that were resulting from the transition reaction are going to combine with a molecule called oxaloacetate. Okay, so when that happens, they form a molecule called citrate. And that's about where I want to leave some of the details, okay? The rest of the process, and there are several intermediary forms in this conversion, but what's happening is the citrate is going to get modified slightly by an enzyme. Then it's going to get modified again, modified. And each of these lines depicts like a different reaction that's occurring. There's going to be changes that are occurring to the molecule. And just recall what I've tried to explain to you guys about multi-step pathways. You have additional control, you have decrease to the losses of energy to entropy. And so it's really important that these multi-step pathways are mediated by enzymes to kind of maximize the energy output for these reactions. But in the end, what we're left with is that same molecule oxaloacetate, and that's going to be ready to grab another acetyl-CoA. So we call this thing a cycle because what we start with is what we're going to finish off with. The acetyl-CoA is entirely consumed by these interactions. So what we started in terms of what entered the pathway, the initial 2-carbon acetyl-CoA, 100% of those carbons are going to be given off as carbon dioxide. Again, converted into carbonic acid in the blood and then respired out in the lungs when we turn it back into a gaseous state. And in terms of products, there are 3 distinct products that we're able to pull off of the citric acid cycle. So for every acetyl-CoA that enters and goes through all of these iterations, we're going to be able to pull off 3 NADHs, we're going to be able to pull off an FADH2, and we're going to be able to pull off an ATP. So what really I really feel like everybody needs to understand at this point is that these 2 molecules are carrying electrons. And that's a form of temporary energy storage. ATP is the target fuel that we're trying to make, okay? So again, the citric acid cycle produces NADH, FADH2, and ATP. Okay, and we should, for every glucose molecule, be able to run this cycle twice, right? Because we have 2 acetyl-CoA molecules. So these numbers would actually be doubled if we looked at a molecule of glucose as opposed to 1 acetyl-CoA. All right, so what do we do with all these NADHs and FADHs we've been acquiring from glycolysis onward? So these guys actually are storing electrons, right? And the electrons are depicted here. We're going to use those electron carriers to go through a series of oxidation reduction reactions. Okay, and remember, oxidation reduction reactions, really all that means is that there's going to be an electron that's going to be passed between molecules. One molecule is going to be oxidized, the other will be reduced. All right, so where this is happening, I hope everybody can visualize here. This is my phospholipid bilayer. I didn't draw the phospholipids in, but these proteins are embedded in a phospholipid bilayer. That membrane is located right here. It's the inner mitochondrial membrane, and we have a series of complexes that are produced utilizing the mitochondrial DNA instruction template, And some of these are really important when we look at evolutionary relationship, because there is a lot of conservation between animals. Like this, any animal really on the planet that goes through electron transport should have very similar instructions for how to make these protein molecules like cytochrome C and other complexes that are part of this electron transport chain. So let's get into details about how this is going to work. Okay, so NADH and FADH2, remember, they're carrying electrons. Those are depicted here. These proteins are, what they're going to do is they're going to shoot electrons or pass electrons to adjacent protein complexes embedded in the membrane. While they're doing that, these transmembrane proteins are going to be able to shuttle protons from the mitochondrial matrix, which is the inside here, to the inner membrane space. And of course, there's the outer membrane is way out here. That's not depicted in this diagram at all. So let's look at this real quick, and I can show it to you a couple different times, but I really want you to understand the pathway and how it's working. So this first electron here is being released by FADH2, and it's going through the proteins in the cell membrane. And eventually, the last acceptor of that electron is oxygen gas. And this is absolutely why oxygen gas is required. Oxygen gas is the final electron acceptor in this pathway. And so if oxygen gas wasn't in the environment, that electron would never be released by this protein complex. And if that was the case, it wouldn't oxidize its neighbor, and that wouldn't oxidize its neighbor. And so you'd have a backlog of electrons. And these compounds, NADH, remember that they are part of a cyclic process. So NADH, that's a compound that's loaded up with an electron. Once it eliminates that electron or releases that electron, it's going to be turned into NAD+. And go back to the citric acid cycle, the previous step, pick up electrons here, and then bring them back over here. Okay, same with FADH2. So if they're not capable or they're not able to be converted back into the FADH+ and the NAD+, then we're going to shut down not only the electron transport chain, but the citric acid cycle. So oxygen gas is absolutely required. And again, electrons are going to be shot off, and they're going to be eventually combined with oxygen to form water. Okay? And so those electrons, in that process of passing them from one protein to the next, they're going to release enough energy to actually actively transport protons in this direction here through these transmembrane proteins. Now, what we've done is we've established a crazy proton, an electrochemical gradient on this side of the membrane. So in the intermembrane space, we have all of these protons that are stacked up like water behind a dam, and they don't want to be there. So if we give them an out, they're going to rush through that out. Okay, so this happens to be the opportunity for these protons to get back into the matrix. And this molecule is called ATP synthase. So what's going to happen is those protons are going to flood through here really fast. Okay, and those protons coming through here provide enough kinetic energy to actually carry out this anabolic reaction where we have ADP, so adenosine diphosphate, and a free phosphate, and we're going to stick that onto the end. So it goes from ADP to ATP. And again, this is the fuel we are looking for. So we have somewhere in the ballpark 32 to 34 ATPs that are going to be able to be generated by the movement of these protons through that ATP synthase. And again, without electron transport, we don't establish this gradient. So there's a lot of pieces to this puzzle. You know, without oxygen, you're not able to move these electrons. With any kind of mutation to these proteins, we're not going to be able to carry out this activity. And all of these things collectively work to completely break down those sugars and manufacture the energy that we're looking for cellular metabolism. So all told, from one molecule of glucose, if we have oxygen available and the mitochondria, we should be able to make around 38 ATPs. Without oxygen, we're looking at a maximum of 2 ATPs net. So the benefit of using aerobic respiration is unreal. Like, it allows us to have such complexity of life, and I hope everybody has an appreciation for the scale of this. I mean, trillions and trillions of interactions every minute of every day. Cells are producing huge quantities of this, and we're constantly giving off heat to our environment because of some of this metabolic activity. And Yeah, it's what allows us to be complex and to stay alive. So beautiful process. Thanks for listening. Have a great day.